Welcome back to our class. Yeah, today we are going to be looking at the process of protein synthesis. What is protein synthesis? So protein synthesis is divided into two words. Number one, it is the protein and the synthesis. When you talk about synthesis, it means manufacture, production of proteins. Protein synthesis is the process by which proteins are made in the cell of an organism to form enzymes, hormones, and the new structures of the cell. So your body is made up of different structures, and these different structures are made up of protein. So it means that if you don't have protein synthesis in your body, then it means that you can't grow, you can't have these hormones, you can't have these enzymes. So the marking point here in the definition is the word proteins are being made in the cell. This process is divided in two different phases. The first phase is called transcription. And when you talk about transcription, it means formation of messenger RNA. Then number two is translation. Translation is the formation of a polypeptide chain. The difference between polypeptide chain and the protein is almost the same, but it depends on how many amino acids are there. We are going to see the amino acids in detail. So how many amino acids are there? If more than 50 and above amino acids, then we call it a protein. But if it's less than 50 amino acids, we call it a polypeptide chain. This diagram shows the whole phase of protein synthesis. This is the nuclear membrane. These are nuclear pores. So we have gone into the nucleus, and then you have gone into the cytoplasm. So this diagram you see, it's just showing you the nucleus and then the cytoplasm. We know very well that the DNA is found in the nucleus. And then this DNA, because it is a king, is the one which codes for the protein, doesn't move out of the nucleus. It's like a king who doesn't move out of the palace. He has the servants, and these servants, they go take information what the king has said. So these messengers, the transfers, they act as servants for DNA, because DNA wants to produce a certain protein. So it will send the messenger, and then the messenger takes the information to the cytoplasm, then the transfer RNA will bring the amino acids. So when they bring a diagram like this, you have to know how to label. That's the first point you need to know so that you can pass this question. The first thing is you have to know that this is DNA. When it is coiled, if you see something coiled, is DNA. If it is too coiled, then we call it chromatin network. Then the DNA is done, which unwinds. It forms the messenger RNA. And then the process of formation of messenger is what we call transcription. Then this messenger RNA will move out of the nucleus via the nuclear pore, and then it goes to the ribosome. You see, these are the ribosomes. This messenger will align itself on the ribosome, and then the transfer RNA will bring the amino acids to this ribosome, and then a chain of protein, a chain of amino acid is formed. Therefore, this process whereby the messenger brings in and then the transfer RNA brings in the amino acid also, the chain is formed, is called translation. So the major difference between transcription and translation: transcription occurs in the nucleus, while translation occurs in the cytoplasm. So you have to know at least the basics of this process. Before we go to the process of transcription, so this is a simple diagrammatic representation of the whole process of protein synthesis. So you expect DNA to unwind, and then when it unwinds, it forms it exposes two strands, but one strand will act as a template for formation of a new strand, and then this new strand is what we call the messenger RNA. This messenger RNA, we saw that it moves out of the nucleus via the nuclear pore, and then it goes to the ribosome. The moment it reaches the ribosome, then it starts the process of translation. Then amino acids are joined together, are brought by the transfer RNA. They are joined together to form a polypeptide chain. So if the number of amino acids become a lot, the more than 50 and above, then it forms what you call a protein. So the protein, it must have is a heavy molecule compared to a polypeptide chain. So now let's look at what happens in transcription. We see in the exam, what do you write when we find such questions, and then what happens in translation. Transcription. Don't forget, this is a recall that transcription occurs in the nucleus. You have the DNA, and then you have the DNA to unwind, and then messenger RNA is formed. Then messenger RNA moves out of the nucleus by the nuclear pore, and then it goes to the cytoplasm. Basically, that's what you're going to be explaining. So the first point, what you see in different color, it means a marking point. DNA double helix. You have to tell us the natural shape of DNA. DNA double helix unwinds. So that's the first step. Number two, it will say that when the weak hydrogen bonds break. Here, when you say weak hydrogen bonds break, or you say DNA double helix unwind, then it unzips. It's the same as saying weak hydrogen bonds break. What's the meaning? If you look at your trouser, you try to open it, you zip down. When you zip down, you are breaking the bond which we are holding the zip together. You understand? So when you say when you unzip, it means that you are breaking down the bonds which are there. That's why we say that weak hydrogen bond break. Or you can say DNA double helix unwind, then unzips. So you get the same mark. One strand will act as a template. There is a major difference between DNA replication and transcription. Here, when you're talking about transcription, one strand is the one which acts as a template, while the other side, the two strands will act as a template. So something which look like a mirror, so that there is one strand, so that the new strand is being formed. Then to form the messenger RNA, one strand will act as a template, as you see it here, and then messenger RNA is formed. It uses free-floating nucleotides. When you say this, you are supposed to specify, because when you say free-floating nucleotides, in the nucleus, there is free-floating nucleotides of DNA and free-floating nucleotides of messenger RNA. So you have to be specific when you come to this. You have to say that free messenger RNA nucleotides. Yes, so these free messenger RNA nucleotides which are floating in the nucleoplasm are the one which we use. Yes, so if you just say using nucleotides, we'll cross it, because we have different kinds of nucleotides in the nucleoplasm. So you have to be specific that free messenger RNA nucleotides. Then the nucleotides, the messenger RNA is complementary to DNA. So it means that where there is A, you're going to have U, and where there is C, you're going to have G. Remember, the messenger RNA does not have thymine. It has what you call uracil. That's why we say A goes with U and then C goes with G. Then the messenger RNA now has coded message of protein synthesis. It means that now it is ready to move out of the nucleus and then go to the cytoplasm, so that it can take the information DNA wants the protein to be manufactured. Yeah, that's why you're saying that now the messenger has the coded information for protein synthesis. Then we are saying that the messenger RNA moves from the nucleus through the nuclear pore, and then it goes where? To the cytoplasm. Where exactly in the cytoplasm? That is on the ribosome. So it basically, this is the general process of protein synthesis, basically that is transcription. So what you see in different color, it means the marking point. DNA double helix unwind, weak hydrogen bonds break, and then one strand will act as a template, and then for formation of messenger using free messenger RNA nucleotide in the nucleoplasm, and then the messenger RNA is complementary to DNA, whereby A goes with U and then C goes with G. And this messenger which is formed has a coded information for protein synthesis, and then this messenger moves out of the nucleus by the nuclear pore to the ribosome. Basically, that's what we want. 6 marks from here. In most cases, you see it as a short question which requires you to write these statements the way you see it. Please don't change. If you change, make sure that the different colors don't change, because those are the marking points we are looking for. And then let's look at the process of translation. Here, let's look at the process of translation.